Good afternoon, folks. Prepare to embark on an otherworldly journey into the realm of chromatin with Michael Meers, a skilled Jedi in the ways of biology.
There we go.
On this special day, May the 4th, has arrived to deliver a talk, or who has on this special day arrived to deliver a talk, Advanced Chromatin Profiling Methods. No, something,
Well,
mapping transcription factor and chromatin dynamics during development. Same thing, but different title. That will take you beyond the reaches of this galaxy. As a young Padawan, Michael was drawn to the mysteries of life, seeking knowledge and wisdom at the prestigious academy known as Duke on the planet of North Carolina. There he trained under the guidance of the Blue Devils, a fierce and determined clan of warriors who embodied the spirit of their namesake.
that's true.
After completing his studies at Duke, Michael's thirst for knowledge led him to nearby planet of Chapel Hill, where he trained with Jedi Master Greg Matera, who was just here on this planet, to unravel the secrets of H3K36 in Drosophila. Legends say that he once used the powerful CRISPR technology to create a hybrid creature, half Wookiee, half Porg, but he remains tight-lipped about the truth of this tale. Nevertheless, Michael's mastery of epigenetics is undisputed. As he continued to hone his skills, Michael embarked on a new quest for knowledge, seeking the guidance of Jedi Master Steve Henikoff at the far-off planet of Seattle, where he developed the revolutionary multi-tag method for single-cell analysis of histone modifications. So now as a Jedi Master funded by a K99R00, Michael has established his own training academy at the Washington University on the planet of St. Louis. There he guides aspiring Padawans in the ways of biology, sharing his insights and wisdom with those who seek to follow in his footsteps. Join us now to welcome Michael. May his words inspire and enlighten us, and may the force be with us all. So I thought it was great.
Yeah, that intro needs a round of applause. That was wonderful, and now I feel terrible that I have zero Star Wars jokes in this talk, but I think Peter maybe provided all of them that we need, so that's great. Yeah, thanks so much for inviting me for this opportunity to speak to you guys. Yeah, I want to tell you a little bit about what I'm interested in the perspective of chromatin structure and how transcription factors interact with chromatin to specify developmental outcomes. And just by way of brief introduction, I'm just going to outline what I'm going to talk about. I'll give you a little bit of intro on my interests and the sorts of techniques we use to get at these questions of transcription factor chromatin interactions. I'll tell you a little bit of a story about profiling pioneer factors during embryonic stem cell differentiation and visualizing their interactions with chromatin. I'll go on to tell you a little bit more about some technology development focused projects I've been working on in single cell chromatin profiling. And finally, just conclude with a few future directions for my lab and where we're going in this direction. But just to preface, to begin with sort of my, I guess, motivating interest, the raison d'etre of the lab is generally understanding how cell fate is determined and how cellular diversity is attained. And in general, we think of this in the context of differential gene regulation, in the sense that we have thousands of different cell types and trillions of different cells in your body that all have essentially the same DNA, somatic mutations notwithstanding, and that DNA obviously gets used in different ways in order to specify different cellular endpoints. And the major way that I'm interested in how this functions is through the function of transcription factors. We know that transcription factors are extremely important in cell fate determination through decades of beautiful work, perhaps the most prominent of which was shown by Takahashi and Yamanaka, who won the Nobel for showing that if you express a certain suite of transcription factors now known as the Yamanaka factors in fibroblasts, you can transdifferentiate and reprogram them back to induced pluripotent stem cell state. But really, this sort of field, I think, goes back even further than that. I was at the Fred Hutchinson Cancer Research Center for my postdoc, and sort of a legendary figure who looms tall in that place is Hal Weintraub, who showed back in the 80s that you could take a single transcription factor, MyoD1, expressed in fibroblasts and specify them to myoblast or muscle cell differentiated fate. So this is all to say that transcription factors are essential coordinating agents of cell fate. And in the intervening decades, we've sort of parsed out a lot of the details of how transcription factors actually accomplish this on the molecular level. This being by sort of binding through their short motif sequences and extended enhancer regions, upon which time they can then recruit different cofactors and coactivators that can then stimulate the activity of RNA polymerase II at constituent genes that are nearby or sometimes very far away. This sort of kind of boils down now in the era of genomics to the molecular underpinning of what would be a genetic switch, right? So transcription factors bind, they're able to activate there's certain target genes, and we can turn targets on and off. And in the context of those switches, we've been able to, with the sort of deluge of genomics data that we've been able to produce over the intervening decades, to produce these broad networks of genetic switches, so gene regulatory networks that specify particular cellular outcomes. And so to that extent, if we know exactly which way the switches are flipped in particular context, then we can better understand exactly how particular cell fates are identified. And that is the end of my talk. No, of course, it's not, because we still haven't been able to do this with sufficient efficiency to actually accomplish that goal. And one of the reasons for this, I think one of the fundamental reasons for this is that the switch isn't actually really a switch in the sense of directly on off. And the reason for that is because DNA does not exist sort of fully exposed in the, as we all know, in the genome, ready for a transcription factor to bind it immediately. Instead, it looks something like this, complexed into chromatin. The core subunit of chromatin being the nucleosome, which wraps roughly 150 base pairs of DNA in a highly stable configuration. Iterate this millions of times in the cell, and you have a fully compacted sort of chromatin environments that can be sort of regulate access to DNA in the cell. And this sort of manifests in different cell types in generally sort of more active regulatory chromatin regions where you have transcription factor binding, you have these cofactors and polymerase. This is associated with sort of particular chemical modifications of histones, such as acetylation, that are associated with a more loose configuration of DNA around the chromatin and sort of more accessible regions. And this is in contrast with other regions of the genome or that same region of the genome in a different cell type that may be repressed. And in this case, the chromatin is more compact. It's more associated with other constellations of chemical histone modifications, such as methylation events. And in the sort of the fundamental problem with the switch model here is that when you're trying to get from A to B, where you have this enhancer that needs to be activated by a transcription factor to activate its constituent gene, this compact repressed sort of chromatin configuration is inhibitory to transcription factor binding. So the fundamental question here is solving this paradox of exactly how do transcription factors initially access their targets in closed chromatin. And I'm going to frame this in terms of two sort of major models for how this paradox could be resolved that have been posited historically in the field. The first of which it kind of invokes this this idea that there's actually a lot of sort of dynamics happening in chromatin where you have sort of closed chromatin regions where a motif might be occluded, but this is these nucleosomes are undergoing constant DNA unwrapping and rewrapping from the nucleosome core. There's constitutive chromatin remodeling activity that's shifting nucleosomes around or evicting nucleosomes, such that in certain opportunistic windows, there may be the availability of a transcription factor binding site that was otherwise occluded by chromatin. And then so in these opportunistic windows, perhaps the transcription factor is able to bind. And you could think of this perhaps as sort of like a dimmer switch where you have a sort of a repressive effect of this nucleosome, but then in the long run, thermodynamics wins out and you're able to, flip that switch to a certain degree, right? Another potential model for how this happens is that there is perhaps actually a special class of transcription factors, and these are collectively generally known as pioneer factors, that actually are able to bind to their target sites in the context of closed and wrapped chromatin. And so in this model, you have a pioneer factor binding to its target in closed or wrapped chromatin, which potentiates some sort of unspecified pioneer activity, which may be direct binding and delamination of the DNA from the nucleosome core, or alternatively recruitment of sort of chromatin remodelers in order to kick the nucleosome out of the way. And that way you get a configuration where this DNA is now accessible and it's available for binding of other downstream transcription factors that may not have this pioneer factor binding mode or ability. And so in this context, this is sort of like if we're extending our metaphor, this would be sort of like a linked switch where like one switch has to be flipped before the next switches downstream can proceed, and they're fundamentally sort of connected to each other. Now there's a tremendous amount of in vitro evidence for both of these models, but it's exceedingly difficult to define which of these is actually the dominant function in vivo. And in order to determine how this sort of parse between these two models, we need a couple things. So first, we need to know exactly where transcription factors are binding in vivo. So we need to have a spatiotemporal map during development. And we also need to know when the transcription factor is binding, what's the chromatin structure there at the time it's binding. So is it actually engaging nucleosomes, or is it just using this sort of bypass effect of the first model? And the way that we have proposed to take control of these like two elements is through a suite of genomics techniques known as chromatin profiling. And so I'll go on to sort of describe a brief history of the chromatin profiling that we do in the lab. So chromatin profiling collectively is the basis behind it is essentially figure out what DNA is associated with a particular protein that you're interested in terms of a transcription factor binding to that DNA or a nucleosome wrapped with that DNA. And you're probably all familiar with chromatin immunoprecipitation sequencing, which is perhaps the dominant chromatin profiling method in which DNA is sort of cross-linked to proteins and sheared, and then you have antibodies that pull down your protein or modification of interest. And this has been extremely powerful over decades to specify exactly where different proteins we're interested in or binding in different cellular contexts. But it has some drawbacks. It has a relatively low resolution due to the fact of random shearing of the DNA not being related to like the biological characteristics of what's actually binding. And since it's an immunoprecipitation-based method, it has relatively high background, and so that leads to somewhat higher sequencing requirements. So we use a separate suite of chromatin profiling techniques known as collectively as enzyme tethering methods. So in this case, we're not breaking apart the cell. We're actually doing everything in the context of the in situ chromatin environment. And it's a little bit more akin to immunofluorescence, in which you're flowing in an antibody, and then that antibody can bind to your protein or modification of interest. And then that antibody is used as sort of a locus point to tether a separate fusion protein that contains an enzyme that either digests DNA directly around where the protein is bound, or otherwise tags the DNA in another way to directly extract or amplify the DNA sequence that is associated with the protein binding. So this has very high resolution because the nuclease and tagging-based approaches are sort of directly oriented exactly around where the protein is bound, and the library preparation is highly efficient. There's no physical pull down in immunoprecipitation, so it has relatively low background, low sequencing requirements. The history behind some of these specific methods that we use begins in 2004, where Uli Lemley designed these proteins these approaches known as CHIC and CHEK, particularly CHEK, which makes a genetic fusion between a protein, a DNA binding protein of interest, and a micrococcal nuclease. So this MNase is a processive endo-exonuclease that digests DNA right down to the boundary of where your protein of interest is bound. And so this DNA can essentially just flow out, and then you can characterize it in some way. Back in 2004, this would have been through Southern blot. Luckily, we've updated that since then. So Gabe Zentner in Steve Henikoff's lab adapted CHEK for high throughput sequencing to do CHEK-seq. And subsequent to that, Pete Skeen further adapted this to a technique known as cut and run, cleavage under targets and release using nuclease. And just to briefly recap how cut and run works, again, we have our antibody to our protein of interest. And in this case, we have a fusion protein between bacterial protein A, which itself binds to the IgG of your antibody, and then you have MNase fused to that. So the MNase can, once it's activated by calcium, digest the DNA directly around where your protein is bound. The chromatin complex diffuses out, and you're able to sequence the transcription factor bound DNA. Okay, so to go back to our question of exactly how pioneer factors are functioning, we decided to use cut and run in a sort of pretty standard in vitro differentiation of human embryonic stem cell differentiation to endoderm. This is a 5-day time course, and we can take 5 24-hour time points here and perform cut and run for a host of different transcription factors and histone modifications. And crucially, a number of these transcription factors are putative pioneer factors whose function is essential to specifying this endodermal cell fate from embryonic stem cells. And you can see here that across this time course, whereas early on you have a high expression of pluripotency factors such as OCT4, as you proceed on, this sort of declines and is replaced by the expression of endoderm-associated factors such as SOX17. So we can do cut and run for a number of different transcription factors in this time course. So here, this is essentially what the data look like. And so if we look at a pluripotency associated transcription factors such as SOX2, if we look at this time course, day 1 through day 5, ESCs being at the top and endoderm being at the bottom, SOX2 is pretty tightly expressed during pluripotency. So you can see the factor binding really strongly early on, and that declines immediately as you go on in the time course. In contrast, if we look at an endoderm-associated factor such as FOXA2, this I'll be coming back to a number of times in the talk. This is sort of your classical pioneer factor that is thought to be able to engage its targets through closed chromatin. And again, this is expressed later on in the endoderm time course. So early on, we don't get binding, and then as we proceed along, we get these peaks of binding of FOXA2 at its constituent motifs and enhancer binding sites. We can iterate this across a whole host of different factors, and what we're looking at here is heat maps where we've collapsed down in each row being an individual locus where transcription factor binding is occurring, and the intensity tells us exactly the intensity of the transcription factor binding in this window around the center of the peak. And across this time course, we can group these into pluripotency-associated factors, binding early, intermediate factors, endoderm-associated factors binding late, and then also invariant factors such as CTCF, which bind throughout the time course. And just to more sort of specifically parse this out, we can look at individual loci within this, and again, see that we get really nice profiles that represent exactly where and when these transcription factors are binding. Since we have data for a whole host of transcription factors, we can actually take the host of transcription factors and time points and cluster roughly 82,000 binding sites that occur for any individual transcription factor at any individual time point, and come to some conclusions about how transcription factors are binding at particular loci across time. And importantly, there are a number of different classes that stand out here. CTCF, again, I told you, is sort of an invariant binding factor. So all of those sort of binding sites cluster across time points, so that across time points, days 1 through 5, we get robust binding at the same binding sites. You can see at pluripotency in day 1, you have coordination of the pluripotency factors. And similarly at endoderm day 5, you get clustering of a whole host of transcription factors, including GATA4, SOX17, EMS, and FOXA1 and 2, all at the same locations at day 5. And finally, you have this this notable cluster that I'm calling all here, where you actually get robust binding of a host of different transcription factors all across the time course, which suggests that there's sort of a handoff from the pluripotency factors to intermediate factors to later sort of endoderm-associated factors, all coordinated at the same loci. So this suggests that there's a lot of dynamics occurring in terms of different transcription factors coordinating how these enhancer activation events are carried across. And I'll come back to this sort of cross-coordination across time later in the talk. But just to get back to the fundamental question of how we distinguish direct from pioneer factor nucleosome binding. So we have really high resolution spatial, sort of spatiotemporal binding data. But in order to get at this question of these two different mechanisms, we need to add another layer of information that we get from cut and run, which specifically is fragment size. And I'm going to tell you a little bit about how that plays right now. So in these two mechanisms, we kind of have two different particles that we can imagine are coming from our cut and run data, one of which would be direct transcription factor binding to the DNA, and alternatively, this hypothetical sort of pioneer factor binding to DNA in the context of being wrapped in a nucleosome. And since I told you that MNase is a processive endo-exonuclease, these two different particles are going to produce two different types of fragments. So transcription factor binding directly to DNA is only going to protect a very small amount of DNA, and therefore we should expect to get sort of these short, small fragments out of these binding sites, whereas these particles, since they're bound in the context of the nucleosome, which I told you wraps roughly 150 base pairs of DNA, we're going to have relatively large fragments associated here. So small fragments being, less than 80 base pairs, large fragments being, around 150 plus. So we can take our data and our spatiotemporal binding data, and we can actually add that second layer on a y-axis of fragment size and produce both single DNA fragment plots where an individual fragment is plotted on a genomic position versus fragment size two-dimensional plot, and then produce average fragment size profiles based on those. And so this is just looking at a binding event at the SOX2 locus, where you can see already we're getting representation of this sort of direct DNA binding motif that's less than 100 base pairs down here, and then is flanked by nucleosomes on either side. So from these sort of average fragment size profiles, we can try to infer exactly what sorts of binding events are occurring across time. So if we extend this across time, we can come up with a hypothesis as to what the fragment size profiles would look like over time and under these two models. So in the site exposure model, where you're getting sort of opportunistic binding, we would expect that direct DNA binding is going to be occurring right from the beginning of the onset of the pioneer factor binding to its target. And so that should start with small fragments and continue on to small fragments along the way towards the end. If we're looking at pioneer nucleosome binding, if it's engaging in the context of a nucleosome, we would expect early on at the time that the pioneer factor is first engaging the DNA, it's going to be binding in the context of a wrap nucleosome, and therefore have large average fragment size, which then collapses down to small average fragment size as the nucleosome is displaced and pioneer activity is enforced. And sort of the first major surprise of this work is that if we look at a canonical pioneer factor, again, FOXA2 here, and we look at all of its binding sites that are represented by small fragments at the last time point and sort of walk them back in time, we can see we get robust representation of both of these mechanisms for the same pioneer factor at different sites. Okay, so this is looking at sort of three different, sort of the day 3, day 4, day 5, or early, middle, late in this case, relative to where the factor is expressed at its last time point. And this is actually generalizable across a number of pioneer factors we looked at. So in comparison with CTCF, which again, we know is bound throughout the time course, where we get very few of these apparent nucleosome binding events. For these other pioneer factors, we look at up to 50% of binding events appear to be mediated through large fragments early on in the time course that then collapse down to small fragments, suggesting that there's some sort of partitioning of pioneer-oriented binding sites versus non-pioneer-oriented binding sites for different pioneer factors that we're looking at. So the obvious immediate question here is, what's the difference between these two types of sites? And the first hypothesis we went to is that inherent nucleosome accessibility may be dictating this. So if you have the site is accessible, then it's going to be oriented via direct binding. If it's inaccessible, then be oriented via pioneer binding. And we can get at this by using this attack-seq accessibility profiling method, which you may be familiar with. Uses a TN5 transposase that directly integrates sequencing adapters into the genome at sites of accessible DNA. So in this case, if we have sort of our pioneer factor inaccessible sites, then there won't be integration, whereas if we have our direct binding sites, there will. So our expected result is that at direct binding sites, we'll have robust attack-seq sort of signal, and that nucleosome binding sites we will not. And then the second major surprise of this work was that that wasn't at all what we found. So if we look across the time course, we actually see that these apparent nucleosome pioneer binding sites versus direct DNA binding sites appear to have the same accessibility throughout the time course. And so there's not a tremendous amount of difference here that might be specifying exactly what binding mode it's using. And this sort of makes sense in the context of this dynamic nucleosome environment, where at any given time, you have the same likelihood of a particular site being accessible or inaccessible. Early on in the time course, it's more likely to be inaccessible than not. But if there's sort of this dynamic shifting that's occurring with nucleosomes sliding around and DNA delaminating off the core, then you may expect that at any given site, you're going to have the same likelihood of that occurring. So that's what we think we're seeing here. And so the question is, what's the next place we go in terms of the difference between these sites? And an alternate hypothesis would be that there's some sort of intrinsic signal that's dictating the binding mode here, and perhaps DNA sequence seemed like a decent place to go here. That'd be an intrinsic signal that might differentiate these. And indeed, if we look at, for instance, coming back to FOXA2, we look at FOXA motif variants where we kind of have this optimal motif that would be, from an in vitro perspective, sort of the optimal binding motif for FOXA. The finding we have here is basically that at direct binding sites, we have a higher representation of these optimal motifs than at nucleosomal binding sites, versus degenerate motifs, which are degenerate at least 2 base pairs in this motif context. We have much higher representation in these nucleosome pioneer factor binding sites than in direct. So the finding here is basically that the difference between the pioneer and the non-pioneer binding sites is that the pioneer binding sites have somewhat more degenerate motifs. This can be formalized if we look at a defined motif score, which is basically a higher motif score means that there's a smaller distance between the optimal motif, hypothesized optimal motif and the average motif, the actual motif. And for FOXA2, we can see that these direct binding sites have higher motif scores than the nucleosomal. And this is, again, is generalizable across essentially all of the pioneer factors we looked at. So the sort of answer we're coming to here is that that there's a difference between pioneer versus non-pioneer factor binding sites that's partitioned inherently by the factor in some way, and that partitioning seems to be mediated through DNA sequence. And just to layer one more thing onto here, and I told you about sort of the coordinated binding that we're seeing across time for different factors. If we go back to those findings here, where again we have this set of binding sites that seem to be bound early, middle, and late by all these sets of transcription factors coordinated across time. If we look at nucleosomal binding sites, we find that nucleosomal binding sites are pioneer factor binding sites. For all of these pioneer factors tend to be more of a constituent of this sort of domain of all sites than the non-pioneer factor binding sites do. So the finding here is basically that not only are the motifs different, but also it appears that there's collaboration between different transcription factors at the same pioneer-oriented binding sites, perhaps to coordinate displacement of the nucleosome. And this we can kind of delve a little bit further here to show that at nucleosomal binding sites for FOXA2, nucleosomal versus direct, we get FOXA motifs represented at both of these types of binding sites. But then if we look at motifs for other factors such as GATA and GATA4, we actually get a higher incidence of GATA4 motifs at nucleosomal binding sites than we do at direct binding sites, suggesting that, again, there could be some collaboration between multiple pioneer factors at pioneer-oriented binding sites to coordinate accessibility of the target site. So just to synthesize all this data, our hypothesis here is basically that there is sort of these dual modes that pioneer factors can employ in order to ensure that their binding sites are bound and become active. So what we're seeing is that at high affinity binding sites, if we have this mechanism of sort of dynamic chromatin where DNA is unwrapping and rewrapping off the nucleosome, in these opportunistic windows where the binding site is accessible, high affinity binding sites are more likely to be bound stably by the transcription factor on its own, and therefore it's able to sort of anchor there efficiently purely via thermodynamics. Now, at weaker binding sites, we think what we're seeing is that the lower affinity here means that in these opportunistic windows, it's less able to anchor there when it gets the opportunity to. And so in order to compensate that, it may anchor on the nucleosome transiently in order to sort of force a more kinetic crowding of these factors to the sites in order to ensure that they become bound. And that may be supplemented by other factors that are also binding in the same place in order to sort of coordinate the sort of co-binding in a way that would ensure that these nucleosomal binding sites become active. And so to sort of further extend the switch metaphor, perhaps this is sort of a capacitor where we're sort of smoothing out the sort of activation energy of a site that is occluded versus not occluded by chromatin at this time. And so these are this sort of fail-safe mechanism is sort of a hypothesis that we're interested in actively investigating in the future in the lab. So just to recap this part of the talk, I told you that cut and run, and particularly the fragment size that we get from cut and run, is very important for being able to infer sort of the protein particles that we're getting out of these profiling experiments to infer exactly how transcription factors are binding relative to chromatin during development. And we can show that pioneer factors engage nucleosomes versus two different distinct mechanisms that appear to be encoded in sort of the sequence-dependent manner and perhaps aided by other factors collaborating. And then finally, we have this hypothesis that pioneer factor nucleosome binding might act as sort of a fail-safe to ensure target activation. So I'm going to pause here really briefly to take a drink of water in case anyone has any questions before I move on to the next part. Yeah.
So are the two different types of pioneers, like that exist, the ones that are in nucleosomes early on and then the ones that aren't? Are they similarly enriched in sort of the defining target genes of whatever differentiation factor?
Yeah. Well, to the extent that the pioneer-oriented binding sites are more likely to occur in these temporally coordinated modules where there's a lot of factors binding across time, those tend to be the more, on average, tend to have more important sort of developmentally oriented, like transcription factors and things in them. Yeah.
When you incorporate other transcription factors, binding sites,
yeah.
Seem like the ones that are most important might be in those regions before you have other.
Yeah, and it's sort of paradoxical in a way because it's there's more sort of developmental transcription factors represented in that cohort, and yet their binding sites are weaker. So I think I could extend this is purely speculation at this point, but I think you could extend sort of an evolutionary explanation where you have collaboration of multiple factors and the ability to anchor on nucleosomes that sort of relax the constraints of these really high affinity binding sites, so that the effect that you see now is that there are multiple somewhat more low affinity binding sites whose fidelity is still able to be enforced through these mechanisms.
Yeah,
but that's
the high affinity binding sites enriched in non-functional.
Yeah, I can't speak enough to, like, I don't have enough memory or data on the sort of like Go terms and things like that to recall whether that's the case, but yeah, I'm not sure. Yeah.
Have you tried, I don't know what data sets are available, like for these development, especially but you try integrating data on every nucleosome position or like turnover, and to see if there's anything there between these two types of mindsets.
Yeah, I mean, so what we've done is we looked at the attack sequence I showed. We've looked at different histone modification profiles with cut and run. We haven't done dynamics being like, for instance, sort of a like metabolic labeling approaches to see what like the speed of turnover at different times. We haven't looked at those data. I'm not sure if those exist for some cell type. I'm not sure if they exist for this particular trajectory. I mean, actually, one of those methods was developed in Steve's lab back in 2010. So we didn't do that, but that would be a really good idea to take a look at that for sure. Yeah.
I'm curious how this compares to, because you said it has very high resolution.
Yeah.
How it compares to ChIP-XO?
Yeah, so ChIP-XO also has very high resolution. And so ChIP-XO is, you get the sort of two strands, like, and on the 3' end, I think, of each strand, you basically get the boundary of the protein, right? The benefit with cut and run is theoretically you're getting both boundaries in the same DNA fragment. So whereas in ChIP-XO, you sort of see one edge, but you don't see the other edge because the other edge is basically used to map. So it's sort of extended out. So in this case, that's why we can actually produce these average fragment size profiles that are informative in this case, because each individual fragment represents an individual protein particle that we're looking at in theory. So from the perspective of bulk resolution, it's probably similar, or ShipExo might even be a little bit better. But from the perspective of being able to sort of parse out these things that double edged sort of thing is important. So yeah.
And here's if you try to complicating their transcription, your pioneer factor, so that you only have these sequence recognition of the, and if you were to do a cut and run on that, would that delay the interaction with the nucleosome kind of stuff?
That is a fantastic idea and something we are interested in doing in my new lab, but we haven't yet. But yeah, I think it's highly likely that elements outside of the core DNA binding domain are mediating some of this. And we have a strong interest in being able to like define those structural elements. So yeah, I'm going to move on to the next section for now, but I will take more questions at the end for any part of this. So moving on and switching gears a little bit. So to the extent that chromatin and gene regulation, as it's encoded in chromatin, is the confluence of hundreds of different proteins that are arrayed at thousands of different sites in configurations that approximate active and repressed, but are very diverse in their own ways. To get a full understanding of gene regulation, in particularly from the pioneer factor perspective, where we want to know exactly where a pioneer factor is binding and the chromatin context around it that can be directly measured to the extent that we can. I have an interest in developing new technology-oriented, so chromatin profiling-oriented technologies to get better resolution of multiple proteins profiled in the same cellular context. And so in this way, if we can get down to a level where during a developmental trajectory, we can see exactly across time how different proteins are being used at different loci, ideally on a single cell level, then we can start to produce really fine-grained trajectories of how gene regulation coordinates successful and unsuccessful developmental outcomes. And so from this perspective, we need a couple of things. We need, again, a single cell chromatin profiling method so that in the same, we're confident that in the same cellular context, we're capturing everything that's happening. And then within those single cells, be able to distinguish multiple different chromatin proteins in the same assay. So to do this, I've adapted a technique that's similar to cut and run. It's known as cut and tag. And I'm sure many of you are familiar with this, but I'll run through it very briefly. It's very similar to cut and run, except in this case, rather than using a protein A MNase fusion, we're now using a protein A TN5 fusion. And TN5, if you'll remember, is the enzyme that's used in a TAC-seq that uses NGS adapters that can be directly integrated into accessible regions in the case of a TAC-seq. Here, since we're using a protein A to anchor it directly at a site of antibody binding, we're now getting integration not in accessible regions, but in regions of actual chromatin protein enrichment. So if we can take for instance, a modification of interest, and then integrate sequencing adapters directly at its site of enrichment via cut and paste DNA transposition. So this is efficient enough that we can profile histone modifications at single cell resolution. So this is a domain of H3K27 trimethylation, and then on the axis here we have each row as an individual single cell with different fragments representing enrichment of K27 trimethyl in those single cells. So we can abstract upon this, and since the really cool thing about TN5 is since we're integrating sequencing adapters, we can actually add kind of whatever we want to those sequencing adapters, including different barcodes that can be paired with specific antibodies of interest, so that when we're reading out our chromatin profiling, not only do we get the position of the protein bound by the actual the genomic DNA, we also get the identity of the protein by the barcode that we're reading out. This method I sort of dubbed multi-tag or multiple target identification by tag notation. So again, if we look at just as a control, if we look at K562 cells and we're profiling, in this case, histone H3 lysine 27 trimethylation, which is associated with developmentally silenced regions of the genome, and RNase polymerase 2, serine 5 phos, which is obviously associated with transcription. These should be completely mutually exclusive in K562 cells, and you can see if we do cut and tag for them, they do occur at very different locations. And now we can do multi-tag either in different cells or in the same cells for these both of these different modifications, read out the identity read out via barcodes, and we get really efficient separation of these across the method in the same cells. So now we can push this to the single cell level, and just to orient you about what I'll be showing you here, we'll take a mixture of cells, K562 and H1 embryonic stem cells. We can do multi-tag for 2 different histone modifications, active and repressive associated, and then do combinatorial indexing to capture thousands of cells, individual cells at once, and then do a dimensionality reduction of chromatin features to two dimensions via UMAP. And so you may be familiar with UMAC dimensionality reduction, that the plots look something like this, where you get different clusters of cell types. But in this case, we're producing multiple different dimensionally reduced data sets, one for each modality here, for each histone modification, and then a merged modality that takes all the data and does nearest neighbor integration to produce a fully dimensionally reduced data set that represents all of the histone modifications that we're looking at. So we can actually link all these. Each individual cell is represented 3 times, once for each modification and once for the merged identity. And then we can use normalized mutual information to identify which dimensionally reduced data produces the best clusters, basically. So NMI of 1 being the best and NMI of 0 being the worst. And this is what the data look like. We profile 22,000 K562 H1 cells or NICs. And if we look at the reduction by the either repressive or active histone modification or the combination of both, then we get really nice separation of these clusters, and including in this mixed cell population where we don't have a priori knowledge of what the cells are, all the mixed cells tend to cluster into these individual clusters here. So we're really doing a really good job of separating cell types based on either individual histone modification in single cells or in a combination. And we can go on to look at individual genes. So this Pol2 subunit has robust representation of active marks in both clusters. In contrast with HOXD3, the HOXD cluster is repressed in both H1 and K562 cells. And if we look at the HOXB cluster, that actually is repressed in H1 cells, but active in K562 cells, and the converse being this gene SAL4, which is repressed in K562 and active in H1. So not only can we get really good separation, but we also get really good representation of exactly what's happening on the gene regulatory level as we would expect it to be happening. So we can move on to try to push this towards a more developmentally oriented context. And here I'm going to show you data from 3 different histone modifications. So H3K27 trimethyl, as I mentioned, is associated with developmental silencing. H3K4 dimethyl is associated with active enhancers and promoters. And then H3K36 methylation is associated with active elongation. So these sort of represent 3 different waypoints sort of in developmental gene regulation, from silencing to enhancer promoter priming and activation to active transcription elongation. And again, we can dimensionally reduce this data. In this case, we're getting 4 reductions, one of which is this nearest neighbor integration. And one of the cool things that we can get from here is if we look at the enrichment of these different modifications, we can actually look at co-enrichment of different histone modifications in the same single cell at the same loci. And it's a little bit hard to see here, but basically we have a spectrum from these active marks, K4 and K36 in blue, to these repressive marks in K27. And then in the middle, we get this yellowish that indicates basically what we would call bivalent chromatin, where we get co-enrichment of active and repressive marks at the same loci. And this has been well established as a hallmark of embryonic stem cells, are highly enriched in bivalent chromatin relative to other cell types. And indeed, if we look at the H1 cells versus the K562 cells in our clustering, and we just look at like bivalent co-enrichment, there's much higher bivalent co-enrichment in H1 cells. If we look at specific co-enrichment between modifications, K27 trimethyl and K4 dimethyl are much more highly coordinated in H1 than K562. But interestingly, we saw the same thing for K27 trimethyl and K36 trimethyl, which traditionally are thought to be antagonistic to each other. So our a priori expectation would be that these two would not co-localize with each other. And we were a little puzzled by this, but we went back to the ENCODE ChIP-seq data and compared it with our multi-tag and found that we could recover all of these events. And the reason we could is because most of these are happening in this configuration where it's not actually occurring on the same nucleosome, but there's K27 trimethyl in one region of the gene and K36 trimethyl in another region of the gene. And what we think we're seeing here is basically the cessation of active transcription, where you still have representation of K36 trimethyl, but then That's gradually diluted by histone turnover and then paired with active catalysis of H3K27 trimethyl to actively silence this gene, we may see both of these marks in the same gene as it's sort of being actively repressed. And then this sort of ingresses into the gene body as you proceed along. So this actually sort of extends our sort of waypoints where we can get even more information that we know sort of about how and in real time sort of these gene regulatory transitions are proceeding. So I'll finish up just to give you a little bit of data on a real sort of developmental transition that we profile this in. Again, this is relatively simple human embryonic stem cells, and this time differentiated to 3 different primordial germ layers: endoderm, mesoderm, and ectoderm. We could profile 3 different histone modifications here. Again, K27 trimethyl, K4 monomethyl associated with active enhancers, and K36 trimethyl once again. And then we can project based on K27 and K4. And a couple important things to note here. So in the combined joint projection, if we do, we can actually do pseudotemporal ordering of the sort of individual single cells on a timeline from sort of these early ESC time points to the fully differentiated time points. And if we do this with our multimodal data, we actually recapitulate all the expected branch points, both between sort of ectoderm and mesendoderm at the beginning here, and then between endoderm and mesoderm later on in the trajectory here. And this is supported by sort of this joint pseudotime that we can look at across time, sort of proceeding as we go down the scale here. The second thing that we can get from this is that if we look at individual trajectories, in this case mesoderm, and we compare this to sort of publicly available RNA-seq data, where in this case we have genes that are expected to turn on across this trajectory, we get this really nice transition again, where we're going blue is more active, red being more repressive, and then yellow be bivalent. Where early on, when these genes are repressed, when these genes are off, they're associated with these repressive histone marks. And then as we proceed, we know from the RNA expression data that they're coming on. And then later on in that time course, indeed, we see the sort of these repressive marks going away and these active marks coming up. So this gives us even more confidence that we're getting really nice sort of temporal trajectories of chromatin enrichment data here. The final thing that we can look at is that within this enrichment data, since we're profiling multiple histone modifications in the same cells, we can actually get the balance of different histone modifications and how they're used in each individual cell, whether there's a sort of repressive dominant or an active dominant function for different cells. And this is parsed out really strikingly if we look at the different trajectories across pseudotime on the x-axis. And then here on the y-axis, we're looking at the percentage of reads from each individual cells that come from H3K27 trimethyl, which is our repressive histone mark. And as we can see, all of them proceed apace here, except for the ectoderm trajectory, which declines really rapidly here to have much lower H3K27 trimethyl represented relative to the other marks. And we can further parse this out in actually, if we look at the dimensionally reduced, so this would be the ESCs only over here, and we project the percentage of H3K27 trimethyl onto these ESCs before they've been committed to one particular lineage or another. And the ESCs that tend to be more ectoderm adjacent are already showing this sort of lower H3K27 trimethyl profile, whereas the mesendoderm adjacent ESCs tend to have a somewhat higher H3K27 trimethyl, suggesting that perhaps there's already some priming towards one lineage or another in the ESC population that's chromatin encoded. And we can look in differences in sort of these H3K27 trimethyl low and high ESCs, and we find that a lot of these differences occur at key sort of like neural and like neuroectoderm regulatory genes. So this sort of tells us that we can get a lot of information about sort of trajectory reconstruction for how gene regulation is proceeding on the chromatin level. And I'm excited to use this sort of in combination with other proteins such as pioneer factors to better understand how they're coordinating development. So just to conclude this part, I've told you that multi-tag is a new technology that we use to profile multiple chromatin-associated proteins in the same single cells. We can look at coordination of distinct histone modifications in the same cells at the same loci in ways that inform our trajectory reconstruction. And finally, this appears to be a promising approach for mapping gene regulatory events in developmental trajectories and getting more information about also global shifts in usage of different chromatin-associated proteins. So just to conclude really briefly on where my lab is going with some of these things, we're really interested in the biology of transcription factor chromatin interactions and trying to define on a biochemical and molecular level sort of the pioneer potential of different transcription factors. And this is going to involve validating functional targets for pioneer factors and defining exactly those structural elements that might be mediating exactly how pioneer factors are able to A, interact with chromatin, and B, specify their targets in the context of closed chromatin. We're also going to continue pushing on the technology sort of analysis methods development space, particularly to use some of these multi-tag and other single cell profiling methods to produce these really fine-grained trajectories in places where we know that, for instance, pioneer factors are making key regulatory decisions as to whether a developmental cell fate will be successful or not. And so if we can use fine-grained trajectories to pinpoint exactly the transcription factor binding and enhance our transition events that are underlying successful versus unsuccessful developmental specification, then we can learn more about sort of the gene regulatory architecture there. And finally, I think there are tremendous applications of this to disease, particularly in cancers, where a whole large proportion of cancers have mutations in key chromatin regulatory proteins. And so to the extent that that's going to change the global chromatin environment, we can that can sort of, when overlaid with this existing sort of pioneer factor binding and gene regulatory network, it can sort of upend these context-dependent regulatory hierarchies and maybe lead to opportunistic regulatory hijacking, either by pioneer factors that are now able to overcome new chromatin barriers, or by non-pioneer factors that can take advantage of chromatin barriers that have been ablated. And so better understanding how global changes in chromatin structure can change very specific gene regulatory outcomes from the perspective of how transcription factors bind, I think could be a promising approach. So I want to conclude by acknowledging all the people that helped here, particularly my lab. We're a small group now, but we're growing, and we're getting some, as I've told everyone, we're getting some real experiments done now, so it's very exciting. All my support at WashU, Fred Hutch, where I did my postdoc, Steve, my former postdoc mentor, and Derek and Jay, longtime collaborators, and my funding sources. And I'm happy to take any other questions you have. Yes. Yeah, it's a good question. Right now, there's a decent amount of cell loss, as there is with any combinatorial indexing approach. So we and others have interest in sort of porting this to more microfluidic platforms like the 10x that are a little bit more efficient on that count. But yeah, currently, I mean, just to give you numbers, like we're profiling 20,000 cells in that one experiment, and the input's more than 1 million. So yeah, it's a decent amount of cell loss, and it would be difficult with precious samples at this point. But we have on the tech development side, we have a lot of ideas to improve that. So yeah. Mm-hmm. Yeah.
Yeah, I think go after the early nesting stuff as well. It seems to me that it should be compatible drop, in which case you could also get multimodal information with transcriptional profiling of every cell individually as well, and then actually reconstruct networks from not just the genetic markers, but also how those execute their functions. And that seems to be the name of the game. Part of what you go after. Absolutely.
Yeah. So the single cell or single nucleus RNA-seq is still, that's your best bet for defining cell types, right? Like that is, it's a mature technology for that purpose. The place where it's a little bit lacking is in these intermediate sort of regulatory transitions, right? And I think that's where chromatin can bring something to the table. But yes, for sure, pairing those together will be the most powerful approach. And people, Ming Ren's lab and other have already produced these methods for co-profiling cut and tag and RNA. And it's, yes, in principle, it we can do that with this too. In practice, there are technical sort of details that make it a little bit more difficult than it seems at first, but yes, we're definitely moving in that direction.
Can I go back to the presentation?
Yeah.
I think I was sort of following up on Catherine's question.
Yeah.
Like, right, he's done a beautiful job looking at the binding
Mhm.
and approach, but then the time that actually comes from relevant. But sort of, at which of these sites are they driving either changes in gene expression or changes in accessibility for other transcription factors to bind?
Mhm.
And is there a difference between that functional readout with this direct?
Yeah, for sure. I mean, as we know, probably most transcription factor binding events aren't essential for getting from cell type A to cell type B. So I think that fundamental question is, are the pioneered binding events going to be more predictive of the more important ones, right? And yes, with this work, I can't answer that question. And I think moving forward, I'm very, as we talked about, I'm very interested in defining the rules of like what makes a pioneer factor. And then from there, once we know those rules, we can deploy those in like developmental settings where we figure out observationally exactly what's important and what's not. I think my approach is trying to take it one step at a time and see where we get. But yeah, I agree though. Yeah. Yes.
It's powerful data you have from the cell trajectory, from the students. Have you considered like predict even predict the cell type from the type of chromatin modifications detected?
Yeah. So we're we can certainly derive cell types from these mixtures, right? But that's a relatively easy problem. The problem is like in complex mixtures or in smooth sort of continuous trajectories. Like can you identify specific branch points and things like that? And I think at this point I think chromatins has the potential to bring more to the table there than RNA and other approaches, but there's also headwinds in terms of just technically sort of the number of reads per cell you get back and things like that. Right now, we're still relatively on the low end of that, but again, once we improve that, I think that's the next goal will be trying to see if we can do as good on that score. But yeah.